Today we are going to check out the eye histology. So first each eyeball acts like a camera. So first lens produces the image of an object that we look at, and next images fall on a light sensitive membrane. We know that is the retina. Retina is the innermost layer. So the next what happens is that retina converts the light images into nerve impulse, and thereby it enters into the optic nerve from here and to other parts of a visual pathway, and finally reaching into our cerebral cortex, and from that is reaching to the visual areas in our cerebral cortex, and cortex is the one where the vision is actually perceived and we get the image. So what are the main parts of the eyeball are? First let us check out the outer wall. The outer wall is made by, you can see the outer wall, it is mainly made up by sclera. So sclera is the one which continues into the posterior 5/6 by a thick white opaque membrane, and anterior 1/6 of the sclera is replaced by cornea. You can see here. So cornea is a convex forwards, and deep to the sclera we also have a vascular coat or uvea. So deep to it we have a vascular coat or uvea, which has some subdivisions that are nothing but the part lining the inner surface of the sclera. You can see this inner surface of the sclera lined by a very thin, you can see this pink layer. This is nothing but choroid. So inner layer is of the sclera. Inner surface of the sclera is thin and it is nothing but choroid. So near the junction of the sclera with the cornea, you can see sclera junctioning with the cornea. At the junction of this sclera and the cornea, we can see here, you can see this vascular coat, that is inner layer of the sclera, is getting thickened and forming a ciliary body, and the ciliary body thus continues to form iris behind the cornea, and behind this iris we have our lens. The inner margin of this ciliary body is continued as peripheral part margin of a pigmented diaphragm. So iris is a pigmented diaphragm, and iris lies between the cornea in front and the lens behind. So you can see lens is behind and the cornea is in the front, and the center of the iris, it forms a pupil. This is the way pupil forms here. So iris lies between the cornea in front. Yeah, we discussed it. And next, the iris, there is an aperture called the pupil. So the retina forms the innermost layer of the eyeball, and the space between the iris and the cornea is called anterior chamber. So this is the anterior chamber, and the space while the space between the iris and front of the lens, so iris and in front of the lens, this is called as the posterior chamber. So these chambers are filled by the aqueous humor. So aqueous humor is filled anteriorly, and posteriorly it is filled by a vitreous body. So the part of eyeball behind the lens is filled by jelly like substance called vitreous body. And so this is the basics of our eye. Now let us start with the sclera, learning histology of sclera. So sclera contains a white fibrous tissue, that is collagen, and also it contains elastic fibers and also connective tissue. In the connective tissue mainly it is abundant in the fibroblasts, and some of the cells are pigmented in sclera. And externally the sclera is covered anterior part by the ocular conjunctiva. You can see here, this is the ocular conjunctiva. So the sclera is anteriorly covered by the ocular conjunctiva, and posteriorly by a facial sheet, or also called as episclera. So posteriorly episclera and anteriorly ocular conjunctiva. Here we can't see the episclera. And next, deep surface of the sclera is separated from choroid by a perichoroidal space. So actually you can see this is a sclera, next to it cornea. So between the sclera and cornea there is a space known as perichoroidal space, and there is also delicate connective tissue present in a perichoroidal space. That connective tissue is called suprachoroidal lamina, or also known as lamina fusca. And next, anteriorly to the sclera it is continuous with the cornea. You can see over here sclera anteriorly continuous with the cornea. So at the corneoscleral junction, this is the corneoscleral junction, and it is also called as sclerocorneal junction, that is nothing but reversing the name, or also limbus. So remember, sclerocorneal junction is called as limbus, and a circular channel is present in it. Here you can see this is a circular channel. This is known as sinus venous sclera. Sinus venous sclera is or also known as canal of Schlem, is located in the sclera just behind this corneoscleral junction. So this is a sinus venous sclera. And next, there is also a triangular mass projecting towards the cornea. You can see here this is known as scleral spur. The next optic nerve is attached to the back of the eyeball. Actually optic nerve passes through here. So you can see this is the optic nerve. So the optic nerve is attached posteriorly to the eyeball, just medial to the posterior pole. You can see this is the posterior pole. Just medial to it the optic nerve is attached. And here the sclera is converted like having many pores in it and forming a lamina cribrosa. So the bundles of optic nerve pass through the perforations of the lamina cribrosa. And next, the sclera along with the cornea is collectively forming the fibrous tunic of our eyeball. We know sclera and cornea together form the fibrous tunic, that is the outermost layer of our eyeball. And apart from providing protection, we know delicate structures within the eye, that is sclera and cornea together, and also they help in resisting the intraocular pressure and also maintaining the shape of the eyeball, and its smooth external surface allows the eye movements to take place with ease. So sclera also provides attachment to muscles that move the eyeball. So this is all about sclera. Next, start about cornea. So cornea is made up of mainly 5 layers. So let us understand by diagram. So you can see 1, 2, 3, 4, 5. So outermost layer you can see over here. It is a stratified squamous epithelium. And the second layer is the anterior limiting lamina. And the third layer is the substantia propria. And the fourth, substantia propria is the biggest layer actually here. And the fourth layer is the posterior limiting lamina. You can see here posterior limiting. And the last, that is the fifth layer, is the cuboidal epithelium. In this outermost layer, the cells in the deeper are columnar, the cells in the middle are polygonal, and the cells in the superficial or outermost, you can see this peripheral layer, are flat. So within this one layer, that is outer stratified squamous epithelium only, we are having this many differentiation, that is deeper layer columnar, middle are polygonal, and the outer one are flat. So this but cells are arranged in a great regular manner. You can see over here. We can't see here the microscopic structures. We are also having sometimes this cornea also have microvilli like structures which are helpful, mainly microvilli, which are mainly helpful in obtaining retaining water for long time in the cornea. And next point is that at the periphery of the cornea, it gets continuous with the lining of ocular conjunctiva, as I shown here, ocular conjunctiva. So at the periphery of cornea, it gets continuous with the ocular conjunctiva. One more important thing is that the corneal epithelium regenerates rapidly if there is any damage due to injury or any other thing. So whenever there is injury, the corneal epithelium gets regenerated very easily. Now we have understood the first layer. Now let us understand the second layer. So we know the second layer is nothing but anterior limiting lamina. So this anterior limiting lamina is also known as bowmans membrane. And this bowmans membrane specialty is nothing but it is made up of fine collagen fibers, and these are embedded in the matrix. So that is nothing but about second layer, that is also known as bowmans membrane. So till now we see first one is the stratified squamous epithelium, and second one is the bowmans membrane or anterior limiting lamina. And coming to the third one is the substantia propria, or also known as corneal stroma. So this is also known as corneal stroma. And mainly the thickness of the cornea is formed because of only this layer, and this is made up of collagen fibers which are also embedded in the ground substance containing mainly the sulfated glycosaminoglycans. So that is important point. So as I said, this stroma have mainly the collagen fibers. So these collagen fibers are belonging to mainly type 2 collagen fibers, and they are arranged in a very regular pattern. They form lamella, and that lamella are parallel to each other within one fiber. But when compared with the adjacent fiber, they are attached they are like in obtuse angles with each other. And main thing, the cornea, you can see the outermost one which is transparent mainly because of this arrangement of regular arrangement of these fibers, and also fact that these fibers and the ground substance are having the same refractive index. So because of this also a reason. And the fibroblasts are present in this substantia propria. They appear to be flattened in vertical sections to the cornea, but also seen like a star shaped on surface view. So some star shaped structures are formed, that is nothing but fibroblasts. They are called as keratocytes or corneal corpuscles. So star shaped structures in the substantia propria of the cornea are nothing but the keratocytes or the corneal corpuscles. Now let's understand about the fourth layer. The fourth layer is nothing but the posterior limiting lamina, or also called as Descemet's membrane. So this Descemet membrane is called as true basement membrane, And important points about that is this posterior limiting lamina sometimes gets continuous, you can understand in this diagram. So sometimes the posterior limiting lamina is continuous with our iris. Then it forms an angle, then it is known as spaces of iridocorneal angle, because between iris and cornea. So this is known as spaces of iridocorneal angle, and it also continues with the iris, forming a ligament known as pectinate ligament. Now let us understand about the fifth layer. The fifth layer is nothing but forming the cuboidal epithelium, that is nothing but endothelium of the anterior chamber. So why this word is its name is because cornea, behind cornea we have our anterior chamber, if you remember. So this is the cornea, this is the anterior chamber where we have remembered the aqueous humor is present. So it is continuous with the aqueous humor in the anterior chamber. So it is called as endothelium of a anterior chamber. So this is the fifth layer called as endothelium of the anterior chamber, and it is contact with the aqueous humor of anterior chamber. And this mainly the cells are adapted for the transport of ions, and they possess numerous mitochondria, and they are united with the neighboring cells mainly by desmosomes, that is occluding junctions. So the cells pump out excessive fluid from the cornea and ensure its transparency. So let us understand some of the important points about cornea finally. So you can see cornea has no blood vessels or lymphatics in it, and it receives the nutrition mainly from vessels around its periphery, but it doesn't have itself, so it acquires from periphery. And the cornea is mainly more rich in nerve supply, the nerve fibers which are mainly non-myelinated, and they form a plexus deep to the corneal epithelium, that too in substantia propria. So here in the substantia propria the plexus, nerve plexus is formed, and the free nerve endings are present in the epithelium, that is our corneal epithelium. So this is all about sclera, cornea, histology. Next let us learn vascular coat or uvea histology. Look at our vascular coat or uvea. So in this we have our choroid, ciliary body and iris, so which form the vascular coat or uvea. It is a inner lining of our sclera. So first let us understand about choroid. So choroid histological structure is, it is having upper, you can see the suprachoroid lamina, and this is sclera, right? Under sclera we have the suprachoroid lamina, and this is the choroid proper, or also known as choroid stroma, and below we have the basal lamina, is also known as the membrane of Bruch. So in this choroid proper is further divided into vascular lamina and also capillary lamina. The capillary lamina is having the choroid, it is also known as choroidocapillaris, and our vascular lamina is having blood vessels. So mainly this choroid stroma is mainly made up of connective tissue, and it is major part is covered by our blood vessels, that is our veins and also our arteries, that is network of blood vessels, and supported by connective tissue. So it may also have some pigmented cells, and this is giving a choroid a dark color. So this dark color is mainly making the interior of eye dark, giving a pigment, and also prevents the reflection of light. So by these two factors it is helping in a sharp vision, that is mainly because of sharp images forming on the retina. So this is about choroid histology. And also remember that the vascular lamina has mainly small arteries and veins, lymphatics, and the inner capillary lamina, that is nothing but this here you can see the choroidocapillaris, as I said. So next, the connective tissue supporting these vessels is called as choroidal stroma, and a part of collagen fibers also contains some melanocytes. You can see some other cells here. This way are coming melanocytes, lymphocytes, mast cells, capillary luminous. Remember that the capillary lamina is not at all pigmented, but all other structures are pigmented. So in choroid, which is not pigmented, is the capillary lamina, and the nutrients are diffused out mainly from the capillaries and pass to the basal lamina of our to provide the nutrition to our outer layer of retina, because choroid is between the sclera and the retina, inner side retina, outer side sclera. So the basal lamina, that is base, so it is connecting with the outer layer of retina, that is our pigmented layer of retina. So it supplies to our outer layer of retina. And let us see about the suprachoroid lamina. This is also known as the lamina fusca. So it is mainly non-vascular, that is no blood supply in the suprachoroid lamina, and it is of delicate connective tissue, mainly containing the collagen, elastic fibers, branching cells, and also containing pigment. So remember, suprachoroid lamina have pigment, and also plexus of nerve fibers is present. So some neurons may also be seen in the plexus in the suprachoroid lamina, also known as lamina fusca. And next, the basal lamina. You have seen here that basal lamina. So it is in the light microscope we can see it, and it is also known as membrane of Bruch. Appears to be homogeneous like a single layer, and however in the electron microscope it is seen to have also having a middle layer. Yeah, you can see in middle, yeah, this yellow color. Yeah, so basal lamina is also having a middle layer, and the basal lamina is having middle layer as we discussed. That middle layer mainly contains the elastic fibers, and on either side there is also a layer of delicate collagen fibers, and the membrane is united outside to the capillary layer of choroid proper, right, and also inside to the basement membrane of pigment cells of retina, that is pigment layer of retina, and the nutrients are passing from the capillary layer to the outer layer of retina and have to pass through this membrane. So the basal lamina is said to provide smooth surface on which pigment cells and also receptors of the retina can be arranged in precise orientation. So by this we complete our choroid. Next, let us see the ciliary body and iris of our uveal tract, or the inner surface of our sclera. Let's understand the ciliary part of our uveal tract. So ciliary body is nothing but the anterior continuation of choroid, right? This is choroid, anteriorly continuation with, you can see the choroid. So anteriorly continued as a ciliary body. Also iris is anterior continuation of this choroid layer, that is second layer. So see, iris is nothing but furthermore extension of our ciliary body, and the ciliary body is connected to our lens by a suspensory ligament over here, and between two iris we have the pupil. So this is some basic. First let us understand, and now let us start with our ciliary body. So it is nothing but a ring-like structure continuous with periphery of iris and attached to our lens by a suspensory ligament. So ciliary body is divided posterior flat part. Posterior flat part is called pars plana, and it is also called as ciliary ring, and an ciliary ring, and an anterior part is called pars plica. So pars plana, pars plica. So pars plica is made up of radially arranged ciliary process. So ciliary body is mainly made up of vascular tissue, that is blood supply and connective tissue, and also it has muscle. The important muscle is the ciliaris muscle. So why we are saying it is important? Because it is used in the accommodation of lens. So ciliaris muscle is responsible for alteration in the convexity of the lens mainly. How you can think the ciliary body is changing lens? Because of the suspensory ligament. Through the suspensory ligament enable the eye to see objects at varying distance from it. So in other words, the ciliaris is mainly responsible for accommodation. So the inner surface of ciliary body is lined by double layered epithelium, which represent a forward continuation of our retina. So the ciliary process are radially arranged ridges forming by fold of tissue. So each fold has core of connective tissue, blood vessel covering of double layered epithelium, and the ciliary folds secrete the aqueous humor. This is important. Aqueous humor, it is in the anterior portion. So this ciliary muscle only form, ciliary folds are only forming this aqueous humor, and they also produce some compounds of vitreous body also. So vitreous body, aqueous humor are formed from the ciliary folds. And next, coming to the iris. So iris is a furthermore extension. It is nothing but the most anterior of the vascular coat of eyeball. It is forming a diaphragm placed immediately in front of lens, and it is peripherally continuous with ciliary body, and center is mainly forming aperture called pupil. So the iris is composed of stroma of connective tissue containing the numerous pigment cells, and also embedded with the blood vessels, smooth muscles, and some smooth muscle fibers are arranged circularly around the pupil and constrict them. So they form the sphincter pupillae. So the other fibers also run radially and form a dilated pupillae. So which are running circularly are forming sphincter pupillae, which are running radially are forming the dilated pupillae. And the posterior surface of iris is mainly lined by our double layer of epithelium, which is continuous with the overall ciliary body. Double layer epithelium. So we have seen the epithelium represent forward continuation of the retina. So the cells of epithelium are deeply pigmented, and the pupil regulates the amount of light which is entering. Pupil is here, right? So it amount of light passing in the eye. So in bright light the pupil contract, and in the dim light the pupil dilates, so that the optimum amount of light is required for the proper vision and reaches the retina. So this is all about uveal tract, that is a vascular coat. Check it about retina. So first let us understand the development of retina. Actually this retina is formed from a diencephalon, that is from brain, diencephalon part. So first the proximal part is formed, that is optic stalk, from where now it is known as optic nerve, and the peripheral and the distal part is formed like a cup because of the lens is growing inside, and only two layered structure is formed at first, and next the other layers started forming. So this is first optic stalk and next the optic cup, and this is known as optic hollow structure. First is known as optic vesicle. So optic vesicle is invaginated by lens so that it converts into optic cup. So first optic stalk, next we have optic vesicle, and next comes the optic cup. So after the optic cup, each layer of cup is made by a single layer of cell, and later it persists as a single layered epithelium that becomes pigmented. So form the pigment cell layer of the retina. So now retina has two types of layers, that is the pigment cell layer, and the other one is a nervous layer of retina. So pigment layer is the one, it is only one in number, but the nervous layer is further divided into several subtypes. So totally in retina we have 10 layers according to histology. So next, in the anterior part of this nervous layer, that is in the anterior part, the both layers of the optic cup remain the single layer. So these two layers line the inner surface of the ciliary body. Ciliary body over here, ciliary body, inner surface of the ciliary body is lined by them. That is forming the ciliary part of the retina. If it is covering the ciliary body, then it is known as ciliary part of retina. And the posterior surface of iris is forming the iridial part of retina. That is the posterior part of the iris. This is ciliary body, this is iris. So posterior part of iris is covered by retina. Then it is known as iridial part of retina. And next, opposite to the posterior pole of the eyeball, we have a retina showing a central region. This is known as central region 6 centimeters. This is known as macula lutea. Yeah, we know this is center of region 2 millimeters in diameter, and as yellow color. Yellow color is nothing but lutea. Lutea name has got because of yellow color, and this is a macula lutea. And in this macula lutea, in turn, we have a small area. This is known as fovea centralis because in center. So it is known as fovea centralis. This is where the brightest and acute, you can say the sharpest vision is seen in the fovea centralis. So here comes our histology. So we have discussed just the basic parts, that is fovea centralis. Fovea is where we see the clearest vision. And next one is optic nerve. Optic nerve is attached to the eyeball mainly at the short distance medial to the posterior pole, as we discussed. So this is the posterior pole, medial to it, short distance we have our optic nerve which is attached in here. And the nerve fibers from the retina mainly converge in this region and pass through the lamina cribrosa. Lamina cribrosa is formed by our sclera, as we discussed before. So when viewed from inside, actually the eyeball, the area of retina is seen like a circular area called the optic disc. So now let's just understand the basic structure of a retina. So retina, when stained by our HND, that is hematoscalin and eosin, it appears the layers become apparent, and we can study the retina by special methods. So it has 10 layers in total. So let us understand. The retina is in contact with the choroid as well as with the vitreous. It is contact with the choroid by an external surface, that is contact with the choroid, and the internal surface is mainly contact with the vitreous body, because in this diagram we can understand. So this is choroid, this is retina, then internally it will let us externally with the choroid and internally with the vitreous body, because here it is aqueous humor and this is a vitreous body. So internally with the relation to the vitreous body. So let us understand the layers of the retina. So first, as I discussed, first we have the pigment layer. So this is the pigment layer you can see here. And next we have the neuronal layers. So what are the neuronal layers? Of course you can see here synapses, because neuronal layers, neurons get synapse, right? So these are the neuronal layers. So first we have the layer of rods and cones. And next we have the external limiting membrane as well as internal limiting membrane is also present. But let us study in order. So first pigment cells, layer of rods and cones, external limiting membrane, and next we have the external nuclear layer, and next we have the external plexiform layer, and next we have the inner nuclear layer. Here plexiform means all plexus means synapses are taking place in this layer. Here only cell bodies and neurons are nucleus are present over here. And the inner nuclear layer, And also we have the inner plexiform layer, again synapse, and the layer of ganglionic cells, and also layer of optic nerve fibers, and internal limiting membrane. And here comes the vitreous, because it is innermost vitreous, and outer we have the choroid. So these are the layers. Let us understand them in detail one by one. So first let us take our pigment cell. So pigment cell is a single layer, right, only containing pigments, and the processor from the pigment extend into the next layer. So next layer, that is a layer of rods and cones. Here we have our rods and cones, that's it. Their rod and cone are present, and their processors are present. Next we have the external nuclear layer. So external nuclear layer mainly contains the cell bodies and the nuclei of a rod and cone. So these are nothing but called as photoreceptors, which convert the stimulus of light into nerve impulse, and each rod cell or cone cell are regarded as the modified neuron, because they consist of cell body and the peripheral process and the central process. So the peripheral process are rod shaped in rods and cone shaped in cones, hence they got the name, but the central process are nothing but their axons. So it extends from the external plexiform layer, that is from external plexiform layer, you can see here, up to the and it synapses with the dendrites of bipolar neurons below. So here we have the bipolar neurons, so it gets synapsed here. And next, external plexiform layer. So we have completed nuclear layer. Next we are going to plexiform layer. So it is nothing but known as outer synaptic, because plexiform synapse takes place only of nerve fibers are present over here, and they form a plexus. So the axon of rods and cones synapse here with the dendrites of the bipolar neuron, right? With the dendrites of their bipolar neuron, you can see here the horizontal neuron is getting synapse here. This is known as external plexiform layer, and the process of horizontal cells also take place in the synapse. And next coming to the internal nuclear layer, that is inner nuclear layer. So here the contain the cell bodies nuclei of three types mainly of neurons, that is we have our bipolar neuron, we also have our horizontal neuron, and also we have our endocrine cells. So let us see them in detail. So first let us see about our bipolar neurons. These give off dendrites mainly, which enter into an external plexiform layer. So this dendrites, that is the bipolar neurons, mainly enter into the external plexiform layer, you can see here. And next they synapse with the axons of rod and cone cells, because those are only present over in external, and also axons that enter the internal plexiform layer, where they synapse with the dendrites of the ganglion cells. So in the internal plexiform with the ganglionic cell they synapse, and in the external plexi they synapse with the rod and cone. So this is the function of a bipolar neuron. And next the horizontal neurons, you can see here horizontal neurons. So these give off the process that run parallel to the retinal surface, running horizontally, and they enter into the outer plexiform and synapse with the rods and cones again, and also dendrites of the bipolar cells. So they also synapse with the bipolar cells, and also synapse with the rods and cones. This is a function of horizontal neuron. Next we have our endocrine cells. So these are the endocrine, these are also horizontally placed in the retina, and they are entering the inner plexiform layer when they synapse with the axons of the bipolar cells, and also with the dendrites of the ganglionic cells, right? So this is a function. So this is about endocrine. And next let us see the internal plexiform layer. So this is about internal plexiform layer. So it is an inner synaptic zone, contain the synapse of nerve fibers, mainly the axons of bipolar cells synapse with the dendrites of the ganglionic cells, right? And next both these process synapse with the process of again internal endocrine neurons or cells. So the internal plexiform also contains horizontally placed internal plexiform cells. And next let us see the layer of ganglionic cells. So it contains mainly ganglion cells and contains the cell body of ganglions. So you can see here ganglionic cell, layer of ganglions, only ganglions are present. And next we have seen that the dendrites of the cell enter the internal plexiform to synapse with the process of bipolar cells and also endocrine cells. So each ganglion cells gives on an axon, forms a fiber of optic nerve, we have discussed. And next the layer of optic nerve fibers, that is our last layer. The layer is made up of axons of ganglionic cells, and the fibers mainly converge on the optic disc, where they pass through foramina, lamina, cribrosa to enter the optic nerve finally and give the message to our brain that is cerebral cortex, where there are visual cortex mainly. And next let us see retinal gliocytes. So microglia, we know like that gliocytes, what are this? Apart from bipolar horizontal endocrine neurons, we also have an internal nuclear layer mainly containing the nuclei of retinal gliocytes, also known as cells of Muller. So cells of Muller are present over here. So the cells of Muller mainly give off a numerous protoplastic process that extend through almost whole thickness of a retina, and externally they extend to the junction of layer of rods and cones with the external nuclear layer, and also the process of adjoining gliocyte meet to form a thin external limiting membrane, and internally the gliocyte extend to internal surface of retina where they form our internal limiting membrane. So this is what we can understand easily. So the membrane separates the retina from vitreous. So the external and internal limiting membranes are sometimes described as additional layers of the retina, that is increasing the number to the 10. And next, the retinal gliocytes are neuroglia in nature. They support the neurons of retina and ensheath them. So probably have the nutritive as well as function, and some astrocytes are also present in relation to our retinal neurons. So this is just about them and the appearance of retina in sections. So how do we check out that appearance? Mainly by hematocycline and eosin. Once let us preview our number of nuclei layer about them. So here we have the outer nuclear layer mainly consists of nuclei of rods and cones, and the cone nuclei are oval and single row, and they are having adjoining layers of rods and cones, and the remaining nuclei are of those of rods. So the nuclei in the inner nuclear layer mainly belong to bipolar, horizontal and endocrine gliocytes, and the layer of ganglion cells mainly of single rows of varying size. So the cell outlines are indistinct, so they stain more lightly than the nuclei in the inner and outer nuclear layers. And next we have the layer of our pigment cells which resemble low cuboidal epithelium. So all nuclei in this layer are of similar size and lie in a row. So remaining layers, that is layers of rods and cones, inner and outer plexiform, and also the layer of optic nerve fibers seen as light staining, and no detail is made out. So the layer of rods and cones may show vertical striations. So this is about, and next let us discuss every cell in greater detail. Let us check out the lens. So lens actually has a capsule outside it, and next it is attached with the suspensory ligament. We know lens is attached with the suspensory ligament to the ciliary body. So next lens has an anterior epithelium which is nothing but continuing as a lens fibers. So actually lens has cortex and also inner nucleus. So cortex have all the newly forming fibers, and the nucleus have all the older fibers. So lens so the surface of lens is mainly covered by a highly elastic lens capsule, and deep to the capsule the lens is covered anterior surface by the lens epithelium, and the cells of this epithelium are mainly cuboidal in nature, and towards the periphery of the lens the cells become progressively longer. You can see periphery, they are becoming more longer and converted into long fibers to form a substance of the lens. So the lens contain about 2000 such kind of fibers. And as I said, the inner nucleus is more pliable, and we also have an outer part called cortex, and both parts made up of number of layers are also called as lamina. So this lamina contain the long lens fibers which are derived mainly from our anterior epithelium itself. So first they are cuboidal, next they are converting into long fibers. So these fibers are mainly made up of special transparent protein called crystallins. So crystalline is the protein which is forming this long fibers. So the fibers are mainly hexagonal in cross section and also have a regular geometric arrangement, right? You can say very regularly arranged, and they are attached to each other by their edges. So their edges are attached by each other. So for this purpose the edges bear the knobs that fit into sockets on adjoining fibers. So you can see these are the edges again attaching to the knobs. So this is into sockets, yeah. And lens is examined from the front, then it looks at having some faint lines. So whenever you see lens from the front, you have some faint lines like this. So they are forming a Y-shaped structure. So you can see here, note the Y-shaped lines on front and back of the lens. So it is showing a Y-shaped structure over here, upright, and the lines become more complex in adult. Actually it is seen more commonly in our infant, that is in fetus we find this. In fetus, these lines form a Y shape upright on front of the lens and inverted at the back, and these lines become more complex in the adult. So these lines are nothing but called as sutural lines. So they're made up mainly of amorphous materials, and the ends of these lens fibers are attached to these lines, and each lens starts on a surface that is such a line and follows a curved course to reach the opposite surface, where it's end by joining another such line. So this is about lens. Now let us start with the accessory visual organs histology. So the accessory visual organs are nothing but the extraocular, that is they are not present in our eyeball, that is above the eyeball, that is you can imagine like eyelids and conjunctiva. Conjunctiva is present inside our eyelids, and also we have our some glands are present, and also we have some lacrimal gland, etc. So these are the extraocular structures or muscles also related with it. So you can take here eyebrows, eyelids, and also conjunctiva, lacrimal gland. So these structures, muscles elsewhere in the so eyebrows, as we already discussed in skin and appendages. So now let us discuss about eyelids. So you can see this is the eyelid structure over here. So you have everything mentioned here. The core of the skeletal muscle over there, that is number 1. So this is the core of our skeletal muscle, that is nothing but the muscle which is present in our eyelid. And next we have a dense connective tissue over here. This is a dense connective tissue, and the eyelid also contain the skin, right, because skin is in the anterior surface and hair follicles are present. So this is the skin and this is the hair follicle over here. And also we have our sebaceous glands. So these are the sebaceous glands, and sweat glands, of course, sebaceous sweat glands. And next we have our palpebral conjunctiva, that is conjunctiva. Conjunctiva is divided into palpebral conjunctiva and also our ocular conjunctiva. So here we are seeing palpebral conjunctiva over here. So next tarsal plate, 8th one is a tarsal plate over here, and next 9th one is a tarsal glands. So this is tarsal plate, and near to it we have this red marked structures. These are tarsal glands. And 10th one is eyelash. We know the eyelash, right? And next we have our ciliary gland. 11th one is our ciliary gland. Over here this is a ciliary gland. And next 12th one is our levator palparis superialis. So this is a muscle. You know, levator palparis superialis. So this is a muscle which is present over here. And 13th one is the accessory lacrimal glands. So first I am giving an overview of this diagram so that we'll understand the structures in detail now. So first let us see the eyelid. So eyelid, you know, it's a basic structure which is present that is extraocularly, and anteriorly, you know, it is having true skin and has small hair, because you know this eyelashes, small hair, and also some sweat glands are association, and the skin is thin. Remember that eyelid is skin thin. Yeah, we know that it's very thin and flexible part. Yeah, and deep to the skin there is also a layer of delicate connective tissue, and it doesn't have fat normally. Yes, it is very thin, so no fat normally. And considerable thickness of lid is mainly formed by the fascicula of the palpebral part of the orbicularis oculi muscle. So the orbicularis oculi muscle forms a thickness, that is mainly the skeletal muscle. As we discuss here, we are seeing the skeletal muscle, right? And next, the skeleton of each eyelid is formed by a mass of fibrous tissue that is called tarsus or tarsal plate. So we have seen here tarsal plate. So this is nothing but the skeleton of each eyelid is formed by this fibrous tissue. And next, on the deep surface of the tarsal plate, there are also series of vertical grooves, that is tarsal glands. And the tarsal glands have seen near to this tarsal plates. So these tarsal glands are also called as meibomian glands. These are present here, and occasionally these glands are embedded with the tarsal plate, because this is near to tarsal plate. Yeah, you can see some of the tarsal plates are also present, and each gland also have a duct that opens into the free margin of our eyelid. So next let us see over here. You can see the tarsal glands are modified sebaceous glands, and they produce an oily secretion at the thin film of which spreads over the lacrimal fluid in our conjunctival sac, and it also delays its evaporation. So next, these are nothing but the modified sweat glands, and forming a modified sweat glands is the ciliary glands, or also known as the glands of Moll, which are present in the knit, especially the free edge. If you remember, if you know the eye structure, right? At the free edge we have this mainly glands, and the sebaceous glands are also present with related to our eyelashes. If you see their structure, this is eyelash, right? So related to the eyelash we have some glands. Those are nothing but our glands of Zeiss. And next we have accessory lacrimal glands above the tarsal plate. So this is the tarsal plate, right? Above this we have one gland that is nothing but known as the glands of Wolfram. And next, the inner surface of eyelid is lined by palpebral conjunctiva. We know this. So conjunctiva is, as I said, it is divided into the palpebral conjunctiva and also ocular conjunctiva. So palpebral so you can see here the conjunctiva thin transparent membrane, and it covers the inner surface of each eyelid, that is the palpebral, and also anterior part by the sclera. Sclera is continuous with the ocular conjunctiva, if we remember in the previous section, and in the free margin of eyelid we also have palpebral conjunctiva continuous with the skin, and at the margin of cornea ocular conjunctiva continuous with the anterior part of cornea, right? And the eyelids are closed, the conjunctiva forms a closed conjunctival sac, and the line along this palpebral conjunctiva is reflected to the eyeball, then it is called as conjunctival fornix. So the line of palpebral conjunctiva reflected in the eyeball, then it is known as conjunctival fornix, and superior or inferior. Next, the ducts of the lacrimal gland are also opening into the lateral of this superior conjunctival fornix. So lacrimal fluid keeps the conjunctiva moist. So our tears are using for conjunctiva to keep it moist, and the accessory lacrimal glands are also present here. Superior conjunctival fornix, also known as the glands of Cross. So this glands of Cross, this conjunctival fornix, this we need to remember. And the conjunctiva also consists of epithelial lining which rests on connective tissue. So over the eyelids, this connective tissue is highly vascular and contains lymphoid tissue, and it is also less vascular over the sclera. So the epithelium lining the palpebral conjunctiva is typically two layered. So it is a superficial layer of columnar cells and deep layer of flattened cells, and then the fornix, that is said, this line where conjunctival fornix over the sclera. The epithelium is three layered, that is an additional layer is added of polygonal cells between the two layers. So the three-layered epithelium changes to stratified squamous at the sclerocorneal junction, as we already seen as sclerocorneal junction. So there are three-layered epithelium at this junction, mainly stratified squamous. And next, lacrimal gland. We know our tear gland. So let us understand about this, our lacrimal gland. So you can see first one is a lumen. It is a lumen in between and of an echinus, and next we have the myoepithelial cells. So these are the myoepithelial cells you can see over here, and next third one is the duct. So duct is one which is having more hollow, more lumen. So that is our duct with some cells you can see here. So this is a lacrimal gland. So it is similar to a serous salivary gland, and it is mainly compound tubulo-alveolar gland and contains a number of lobes which drain about 20 ducts. So this is important. And next, the lacrimal gland is distinguished from serous by mainly the echinae are larger and have a wider lumina. Yeah, right. And next, all cells appear to be of same type. They are the low columnar in shape and stained pink with the hematocyanin, and using the common stain we use. And the profiles of the echinae are often irregular or also may appear elongated. And next, the walls of adjacent echinae with a lobule may be pressed together, there being very little connective tissue between them. So very little connective tissue between them, and however the echinae of different lobules are widely separated by the connective tissue. And as the myoepithelial cells are present as in salivary glands. Even in salivary glands we have our myoepithelial cells, and the small ducts of the lacrimal gland are lined by the cuboidal or columnar epithelium, and larger ducts have two-layered columnar or also pseudostratified columnar. So these are last cells you can see, they are lined by the columnar or also pseudostratified columnar epithelium. And electromicroscopic study on human lacrimal gland reveals the secretory cells of several types, including mucus and serous. So it may contain mucus and serous, both cells are present, and the gland is mainly innervated by parasympathetic fibers, that end in a relation to the secretory cells or also as well as myoepithelial cells. So the sympathetic nerves are also present, but their role is uncertain. So parasympathetic and sympathetic both are present, but sympathetic role is still unknown in the case of lacrimal gland. So by this we complete eye histology.